We're live, and this is This Week in Science. We're ready to begin our show in just a moment. Just want to let you know that you're watching This Week in Science, the weekly science podcast, and this is the live broadcast of that podcast recording. Not everything may be in the final podcast, but this is here, all of it right here, right now. Welcome to the show. Co-hosts, are you ready?
Ready, Freddy.
Check.
Sound check, Justin. Say some words.
Checkity, check. Checkity, check.
Checkity, check. We're here, ready to roll. Kevin Unique in the chat room is Groot, and Shoe Brew is not here, but he is here. Okay, time to start the show. Hitting the button, ready to go. In 3, 2. This is TWIS. This Week in Science, episode number 825, recorded on Wednesday, May 19th, 2021. How to use your memory with science. Hey everyone, I'm Dr. Kiki, and tonight on the show, we have lots of things to fill your head, like iconic sounds. Oh wait, I said it wrong. I'm Dr. Kiki, and tonight on the show, we will fill your heads with memory, iconic sounds, and googly-eyed deterrents. But first...
Disclaimer. Know your enemy. You may not think you have enemies, being that you are such a nice person and all, but they don't see it that way. No, they see you as the enemy. And by default, that means you have an enemy. And are an enemy. How do you defend yourself from this fiend? First, you must know your enemy. You will know them by their words, chosen to influence, not inform. You will know them by their actions, spreading fear, not facts. You will know them by the constant deflections away from knowledge, away from truth, away from reason, empathy, and compassion, and away from the most important strength of our sentient modern minds, our knowledge of reality. The only thing now standing between you and the enemy, the only thing that can preserve your freedom, your intellect, your PIN number, and protect the health and safety of your family is This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. 825. Whew. That's quite a several. We've been bringing the science. We'll continue to bring the science every week. And tonight on the show, we have some fabulous stories lined up. I've got stories about, what do I have? I have stories about iconic sounds, repairing hearing, and thumb brains. Thumb brains. What did you bring, Justin?
I've got In Search of Life on Other Planets, Teachers Don't Care If You're Smart, Why Horses Are a Thing You've Heard Of, and How to Not Forget Things.
I don't want to forget things. I want to remember things. This is going to be the TWIS's helpful handy tips in the Justin section of the show. Blair, what's in the animal corner?
I have those googly-eyed deterrents you mentioned. I also have 2 new ways of breathing. One involving bubbles and one involving your rear.
Yeah, we'll get to
my
that.
Ear? Did you say my ear?
Sure.
Well, posterior to that,
Yes.
we need to tell everybody that this is This Week in Science. And if you are not yet subscribed to the show, you can find us all places that podcasts are found. Look for This Week in Science. We're also on YouTube, Facebook, and Twitch. Our website is TWIS, TWIS.org, twis.org. Are we ready for the science? To dive in? Okay, let's bring the science. Okay, I just have to get it out of the way, top of the show. Just talk about it before we go any further. The natural formation, the Darwin's arch, or the arch of Darwin in the Galapagos Islands has collapsed. And a group of scuba divers who were on a boat actually saw it collapse. It apparently collapsed due to natural erosion causes. And instead of now being still called Darwin's Arch, it's now potentially going to be called the Pillars of Evolution, which I kind of like. It's got a nice ring to it. It's got a nice ring. The pillars, it's now 2 rock towers that come out of the water instead of a nice arch formation. However, it just got me thinking a little bit about humans' effects on things. Dan Rather made a post about this arch falling on Twitter, and I replied, "Ha, finally something that humans didn't have anything to do with." But in thinking about it, I was like, well, actually, erosion has probably increased because of our effects on climate change and increases in storms and heating and wind. And so we probably helped it along just a little bit in the last 100 years. But I'm just going to, stop it right there. Other news. China landed a rover on the surface of Mars. It has a mission to look for fossilized microbial life. That's what it's looking for. This makes China the second country in the world to land a rover on Mars.
So
Only
wait
the
a second.
second.
So it's looking for fossilized? So
Yes.
it's basically just got a camera.
It's got a camera, and and rocks. It's gonna be. It doesn't have rocks. It's gonna be looking for rocks. It's gonna be drilling. It's gonna be
has
digging.
A drill. Okay, it has a drill.
Yeah.
It'll be digging, and it has a
It's
camera.
gonna be driving around, checking things out.
It's going to go find Curiosity, shake its hand.
Yeah. Yes. So new locations on Mars being explored.
Nice.
Yeah, by Chinese robots. Now, additionally, this is my last short entry story just to get it off because these are just stories I didn't need to talk about a lot, but I loved them so much. There are over 50 billion birds on the planet. There's at least 6 birds to every human being on the planet, according to a recent survey that took place over the last decade involving citizen science efforts to be able to report on bird sightings. They looked at something like 92% of all the recognized birds on Earth that they were cataloging and estimating, because of course we couldn't actually count every single one. There are estimates being made. These estimates put house sparrows in the billion bird club. There's over 1 billion house sparrows on the planet. So every time you go sit outside at a restaurant and you're like, oh, look at that cute little house sparrow. There's a reason you're seeing house sparrows. There's a lot of them. They're very successful, along with European starlings, ring-billed gulls, and the barn swallow. Very common birds worldwide, all in the billion bird club. And
I would have assumed, perhaps erroneously, that chickens were the most populous bird.
That is maybe something they did not take into consideration in their count.
Yeah, they're probably counting wild birds.
Wild birds, yes.
Yeah.
Wild bird sightings as opposed to the factory farmed kind.
Yeah.
Yeah, but I wonder actually if we were to look into the number of animals, number of farmed birds, how many would be in there? I bet,
I bet
definitely
it would still be chickens.
chickens.
Amongst the farm birds, it would be chickens for sure.
An extremely quick Google. There are 25.9 billion chickens on the planet Earth.
Okay, there we go. So they're well in the billion bird club.
That's over 3 chickens per person.
Is that enough? I feel
We are
like
set.
it's not enough.
I mean, everyone can have mine, so I think you are okay.
You're set.
Still
you
feel a little
you
light.
get your chicken.
Still feel a little
More
light. Even
chickens.
with all 3 of your chickens, still feel a little bit light.
Everybody gets at least 3. Come on, everyone gets their 3 chickens. I'm not going anywhere without my 3 chickens.
It needs
Except
to already
maybe,
be in the pot.
yeah, but maybe I'd be finding other kinds of life if I lived on a different planet. Justin, you want to tell me about life on other planets?
Oh, that's sounds like an intro to a story about scientists searching for extraterrestrial life.
Yeah.
And we have talked about this from time to time on the show. Life extra elsewhere might not look like life on Earth. It might be different. It might have slightly different constituents. So if we have sensors that we've designed here on Earth with our expectations of what life should look like on Earth based on our one-off experience, we might be missing life elsewhere because our sensors aren't looking for how it could possibly be different. This is a new study by a joint Japan and US-based team. This is led by researchers at Earth Life Science Institute at the Tokyo Institute of Technology, and they've developed a machine learning technique that can assess sort of complex organic mixtures using mass spectrometry and sort of reliably classify them into a biological or abiogical category. And they took a bunch of basically what they did is they've fine-tuned this with a bunch of things that they like built in the lab versus naturally occurring organic compounds that are associated with life to some organic compounds that haven't been associated with life. And they've gotten it down to about 95% accuracy using this mass spec, which doesn't sound like sounds pretty good to me, 95%. But if you get that 5% false positive or missing 5% out there in space, that could be a problem. But they basically, right now, the only ways that we are currently looking for life elsewhere, we are listening to radio signals from advanced intelligent civilizations, which we should also tell them we are now on YouTube and Twitch. So it's not just radio. They can look other places. Also,
You can find us all sorts of places.
All sorts of places. They also look for the differences in atmospheric composition around planets and stars. And now we're, like you said, we're on Mars, digging in the dirt, looking to pull samples directly from the soil to see if there's organic compounds. Curiosity rover suggested that there was organic compounds on Mars. It's not exactly evidence of life, but it does sort of tell you that there is possibly the makings. One of the things that we have learned is that while we have life on this planet that started in a particular way, it wasn't the only way that it could have formed. There are slight differences that the life could have taken at the outset. The fact that we have life that all seems to be from the same origination point, as again we've talked about on the show, is likely because once any kind of life got established, the niches fill up, and then anything else trying to come along later after all of these sort of biological niches are filled is immediately starting at the bottom of the food chain. Mm-hmm. And so trying to build up from the first step of life on a planet that already is filled niche, has all the niches filled with predators, exceedingly difficult. Which is why perhaps the only way that life has formed on planet Earth is the way that it happened to have formed, not because that's the only way life can form anywhere in the universe, but because that's what happened first here. And once it starts, kind of hard to have a second branch or another type of life sharing the same planet, starting from
right. Okay, so if I'm understanding this correctly, then it's the reason that we don't have multiple different life trees of different forms is that basically one tree took off, started using all the resources, taking control of niches, competition got going, and there's just no room. Something else would have to come in and outcompete to be able to make it, and
it would have to outcompete. Sort of like if you were a newborn, and now you have to take on every MMA fighter, right, who's in the league, just to get a shot at being noticed. I mean, because you have to that every part of that step of that organic life's evolution has to beat out whatever is there.
Yeah,
and it's too much of a competition. When life starts first, you have a whole planet to expand into, to find whatever resources are there. You can differentiate and tackle it in all these sort of different directions. And every time you arrive at a new resource, you're the only one. You're the only one even using this resource. Yeah. Right. So that appears to be sort of what has happened on this planet. So they've created this system.
This just an aside, it still just makes me think about, why have we not seen, or where is the evidence for other kinds of possible starts that never got the start?
Yeah. And it's because they just wouldn't make it. They wouldn't have room.
They wouldn't have made it. Okay.
So no, and the thing is, it's you don't have to go backwards. It could happen tomorrow. It could have been a month ago that a absolutely new form of organic life began on planet Earth somewhere. Possible.
And then it died.
And then it got eaten. No, then it was eaten. It's like, I like your chemistry. I like your chemistry. Right. Let me use that to fuel my well-established chemistry on this planet.
So I feel like you could follow me here. I feel like you could look at this kind of like in an analogous way to how you use kind of field development to talk about evolution. Like you see a similar progression, and you can apply it to this larger concept. So we have multiple domains or kingdoms or whatever you want to call them, depending on when you took biology. But you have these different types of life on our planet. And yes, they are all based on similar building blocks, which I get is what this is really about, right? Mm-hmm. But still within that, these different domains or kingdoms are so different from each other.
Yeah.
So I think that you can apply that understanding to the universe and understand, because just like Justin's saying, the reason that plants dominated whatever they dominated, if it could be the intake of CO2, they dominated that because they were the first on the block to do that. You could see that kind of in a meta view of the universe where different types of organisms popped up and they were the first to this specific type of niche. So like I always, this is like the thing I always bring up on the show is, okay, but why are we looking for that? Like life could be anything.
But I mean, it's just, it goes to show that the way that organic chemistry works on our planet, the way that life supporting chemistry works on our planet is the way that it got moving fastest first, and then runaway competition for resources. Push, pull, push, pull, push, pull all the way.
And basically what they've done here is they've trained the computer algorithm to detect within a mass spec signature of what compounds are available. They've trained it to identify complex, what they're calling prebiotic chemistry. So figuring out what those sort of, having already figured out what those other precursor chemistries might look like, what other combinations could have been possible, seeking that within the mass spec. So it's a little bit tiered. Very interesting. It might be able to categorize places to look for life a lot easier, a lot faster. This is something that could be very easily mounted onto a probe that's traveling through deep space and taking analysis of things and sending it back. It's not a high resolution, high power necessity to run mass spec. So it's a nice tool that we can add. In other news, UFOs have been seen daily by our government, and they're just now getting around to telling us about it. Have you heard about all of this the UFO stuff in the news?
I've been avoiding it.
I was too. And then there was this interview with Obama where he's like, yeah, no, we don't have aliens. I asked, and we don't have them locked up anywhere. But there is stuff flying around that we have no idea what it is. We get those reports all the time. What?
Yeah, but that could be a satellite from another country that we don't know about, right? It could be space garbage. It could be any number of things.
Unidentified flying object means classified. It means unknown to the government, and they're not going to tell you about it, or unknown to the government, and they're just going to go, oh, we don't know whose that is or what that is. It's unidentified. That is it. It doesn't mean the A word.
No, but we will be reporting on this. We will be reporting on this because sometime in the next month or so, they're going to do apparently a government report, transparency thing, where they're going to dump a lot of this has been classified encountery stuff for public consumption. Yep.
It's coming. I remember they said that was going to happen this year. So
we will talk.
And that was because all those people rushed Area 51, right? Isn't that what?
No.
This is a massive tangent. But anyway,
because they accidentally declassified some 3 videos. So then you have reports of a pilot, which is just whenever I've heard anybody describe a thing, you're like, that's the person saying something that's meaningless. But it's with video, it becomes very interesting. So anyway, we'll talk about yes, you're right. That is a tangent for a future show. But I
thought you had a story for that one.
No, this was the story. It's the end of this story, which is we're looking for alternate molecular signatures for the possibility of life on a distant planet through a mass spec high tech. Meanwhile, they're just buzzing. There's UFOs buzzing airplanes off the coast of San Diego. And we're like, should maybe look at that too.
Well, there is a lot to be looked at our own planet. Yeah. 'Tis true. Especially googly eyes. I like googly eyes. Do you like googly eyes, Blair?
Yes, I like googly eyes, but do you know who doesn't like googly eyes?
Who?
Burbs.
Oh, no.
Yeah.
Burbs.
A team of researchers affiliated with several institutions in the UK and one in Estonia have worked together to create a buoy that will repel seabirds. Why? Because they don't want stuff pooped on? No. It's because, actually something much sadder, seabirds are caught in gill nets all the time. So literally hundreds of thousands, some people think over 500,000 seabirds a year are drowned when they are caught in gill nets, because that's their whole thing, is that they dive quickly into the water to snatch up fish. And a gill net is designed to be invisible in water. So, and gill nets catch fish. So then the seabird goes in and they're like, oh, a fish. Mm-hmm. Then they're stuck in a gill net and they can't breathe underwater. So that's a problem. Aside from just getting rid of gill nets, which would be great, researchers are trying to figure out how to save seabirds at places where gill nets are erected. So to start with, seabirds, like many other creatures, avoid eye contact with other creatures. And so the googly eyes they found were a really good deterrent. And so they put them on the small pole, attached it to a buoy, and then on the top were these big googly eyes. They were big enough so that birds, even with birds with poor vision, like geese, I didn't know geese had bad vision. I'm going to tell them next time I pass them. You can't even see me right. Geese, even geese can see them. And so they kind of scare away these birds by looking like they're looking right in their eyes. And the waves that make the buoy bob back and forth give them this looming effect. Plus, the little the centerpiece of this thing makes it turn with the wind. So then as it turns slowly, birds in every direction get looked at by these googly eyes. I think this looks a lot like Johnny Five. I don't know if
it does a bit. Yeah.
Mm-hmm. Yep.
Yeah,
Short
Johnny
Circuit.
Five from Short Circuit. Yeah.
Yes. So anyway, they selected several sites near the gilt nets. They counted how many birds approached and how many attempted to fish. And then they set up their googly-eyed buoy. And then they counted birds. And over the course of 62 days, they found the number of birds that tried to catch fish near the gill nets dropped by about a quarter, about 25%. Not great, but substantial. That's a lot if you're talking about hundreds of thousands of birds. And that was for up to a distance of 50 meters in every direction. And what's really interesting is they also found that the birds were less likely to fish near the buoys, even if the buoys had been removed, for up to 3 weeks afterwards. So they were like, I don't know, that thing's going to come back. So that's
a pretty good deterrent.
Yeah.
Keep them away.
Absolutely. Also, just don't put up gill nets, please. And if you're buying seafood, if you can buy sustainable seafood, usually that means it was not collected via gill net. So you can look into sustainable seafood also. Anyway.
Fewer gill nets mean that we need fewer googly eyes to deter birds.
Yes.
I think googly eyes look good on everything anyway.
It's true. Yeah.
This should be on more things.
And if you have ever had the opportunity to find the large dinner plate size googly eyes to apply them to things, those are fun.
Yeah.
I know Kiki and I have both been in a home where everything has googly eyes on it. And it is just so satisfying. And if you're listening, hello.
and we're not just saying that because we are backed by a big googly eye.
Yeah, Big Craft has us.
Big Craft. This has got to be. That's where the big money is, so that's where we go. And now we pander to Big Craft.
Yeah, I gotta put a call into Joanne later. Anyway.
Oh, tell her Michael says hi.
She wouldn't take his call. Anyway.
Oh my goodness. That's because she's out running around with the Hobby Lobby. Okay, now.
And Beverly. Don't forget her. Anyway.
And Beverly. Yes, and Beverly. I'm going to move away from sight, the sight of googly eyes, onto the sound. Well, if you...
Of googly eyes?
No, not the sound. We'll get back to the sound of things later. But for people who are born congenitally deaf, there is no current way to give them their hearing back. If you're born deaf, your brain is going to develop without hearing. And for years, there was this idea that the proteins involved in the inner ear, there's one specifically called otoferlin, that is being studied by researchers at Oregon State University in biochemistry and biophysics. These researchers have been checking out otoferlin, and this protein was known to be involved in encoding hearing information. But the experiments that they just did using gene knockout technology, they were able to increase otoferlin's activity or get it out of the cells so that it wasn't there. And they could see how the hair cells, how the cells of the inner ear developed with and without otoferlin. They were able to determine that this protein that had only before been thought to be involved in information encoding, that sound transferring the vibrations into nerve signals, that is actually involved in sensory cell development. And without otoferlin, the sensory cells mature less quickly. They don't mature as well. And so now they're trying to figure out how they can use a smaller form of the gene, a truncated form of the gene, to be able to introduce it as a new mutation, like to using CRISPR, introduce it to people's ears, maybe to young children's ears before they've passed the plastic period of sensory development and hearing development, and potentially fix the otoferlin protein in people who are congenitally deaf. Now, they haven't done anything in humans yet, but in their study that was published in Molecular Biology of the Cell, they tested whether there's a portion of the protein that's necessary in being able to get this new mutation into the sensory cells. And so they did find that there's this particular transmembrane domain, and a transmembrane domain is a part of the gene that goes across the cell membrane. So there's part of it outside and there's part of it inside the cell. So it's definitely involved in some kind of cross-membrane ion channel transduction, some ion movement across the membrane. But anyway, they're studying it in zebrafish at this point. They're transparent and they don't have to be in big tanks. They just have to be in these small amounts of water. And they were able to find that a lack of the membrane domain led otofurlin to properly link in the way that it was supposed to link the neurotransmitter to the cell membrane. And so less neurotransmitter was released. And they say, our study suggests otofurlin's ability to tether the vesicles to the cell membrane is a key mechanistic step for neurotransmitter release during the encoding of sound. And so this whole study potentially is going to lead researchers on a better understanding of how hearing develops in the ear, in the brain, what proteins are involved, how they're involved, and potentially be able to treat mutations in otofurlin that lead to deafness at some point in the future.
Neat.
Yeah, so it's very interesting. I'm super excited and stuff like that. The idea that very isolated genetic modification could take place in the future, where maybe it's eardrops or maybe it's just a small procedure that where doctors go into the inner ear to apply the treatment, and suddenly you have a fixed cell. Suddenly you're able to turn vibrations into sound, into those nervous signals that your brain can then pick up and translate. Yeah, so it's very exciting. Down the road, nothing yet, but it's incredible information.
Very cool. I seem to have acquired the lighting director from the film Casablanca.
That's fantastic.
As you can see, the lighting scheme of backdrops made of shadow. As there's this very bright light being reflected actually off an entirely...
It burns.
It's not even direct. It's bouncing off of a building right now. It's crazy.
That's a lot of fun.
This next story is kind of is also fun. What makes a great student, would you say?
What makes a great student?
Yeah, what makes somebody a great student?
It depends on the teacher. Objectively,
you're not in school anymore. You don't have to keep kissing up to teachers.
Okay, all right. I think asking questions is really good. I think that
and engaged. If the student is engaged, is paying attention, is curious, and wants to gather more information.
Okay.
Yes. I mainly mean because some teachers hate it when you ask too many questions.
Is that right?
I know from experience.
Okay, so what's the answer?
Is it the agile mind, able to race ahead of the lesson and pull conclusions from the content as if the student were in conversation with the ancients from whom the lessons of knowledge were derived in the first place? Nah. Is it this spark of enthusiasm you're talking about? The student shows for the work, their ability to work well with others in the class, and the mark of being prepared to be a good future employee once they graduate. Nope. That's not it. Is it the mind for being academically efficient, getting good grades, and knowing how to operate within a read and regurgitate system of education? No, that's not it either, according to, at least not in the eyes of typical university staff, according to the Journal of Educational Review. The ideal student is punctual, organized, and works hard. That's it. That's what they want. They want you to come in and do the schoolwork and be on time and have your stuff together when you get there.
And this is why my son always gets comments at the end of every quarter.
Ooh.
Your child is disorganized. Seems to have issues organizing himself and getting his work in on time.
Yeah. How did you do on the test? Did you pass the test?
Yep.
Okay, then shush.
It's fine.
Shush. His job isn't to make your job easier. His job is to know stuff.
Mm-hmm. To learn it. But yeah, I get at a certain point, if a student is organized, that means that they are putting the information together in an organized way.
No, not necessarily. So,
if I go back to school, I just need my trapper keeper and to put all the papers in the binder right binders, in the right folders.
They want you organized. They want you to show up on time, hand everything in, formatted the way they want it formatted, and with, stapled properly or bindered, 3-ring bindered properly, or however they want you to submit the work. They want you organized, have the ability to, print out a document or make a copy of a thing if they didn't have time to copy it from the text. Whatever the thing is, this is the result of a new study that involved a survey of 1,000 students and staff at British universities, as well as a focus group with 132 members of both of those groups. Participants were asked to write down the top 5 and least most important characteristics of an ideal student. Interestingly, that list was slightly different between the two groups. The academic skills, employability skills, and intelligence and strategic approach were ranked in the bottom 3, actually by the staff and the students. Although the students did rate employability skills much higher than the teachers, presumably because they may want to get jobs as a result of their education.
So you said these are university professors, right?
No, university is it says university staff, but I'm assuming it's teachers, because why would you be talking to the admissions people?
Right. But this is my follow-up is I want to know what the student body size is of these universities and what their class sizes are. Because if you are in a class of 20 students, the student who is asking questions, who is engaged, who has an interesting conversation around the topic you're discussing is going to pop out and way more than the person who's organized. But if you are assessing your students based on 200 essays you've collected from your lecture hall, and you get almost no interaction one-on-one with those students throughout a semester, your rubric is very different on
how
which
easy is
it
it to make a comment based on how organized they were in formatting their paper or whatever.
Yep.
Yeah. I'm still upset that the just being smart doesn't
cut it. Nope, you got to work hard.
Well, I think
what is smart without using it? You got to use it correctly. Come on.
To be a good teacher, you can't just teach to the smart kids.
Nope.
That's like teaching 101, right? If you have the kid that jumps to the end of what you're talking about, you've lost half the class. So that's not beneficial to everyone else who's there.
Yep. Teaching is about getting everybody through it, trying to get everybody to understand it.
Yeah. And
regardless of their starting intelligence.
Yeah, and that's the part of it I don't like. That's the part that's always
you're not a
me.
teacher.
Yeah, this is why I'm a terrible student, apparently, too, is that the organized part is my lowest priority, and being able to say something smart is my highest priority, and so I've got everything backwards from the way a teacher wants it. But it's also I guess, fair to say that the teacher's job is to feign interest in what's going on in somebody's head. And so the quicker that they can check off a comment that's applicable to that brain, the easier their job is, and the quicker they get through their day and can go and home and drink alone.
I don't know. I think you're oversimplifying just a little. I know we're in the quick stories here, but just from my experience in informal education, I feel like having that conversation and understanding that somebody gets something is so important and so rewarding. But if at the end of the day they don't care enough to put two words together on paper to explain what they've learned, it's kind of insulting. And I think that's part of what this is about too, is about valuing the professor's time and
communication both ways. Education takes
absolutely.
Engagement. It takes involvement. A teacher's not just going to put information out there and the student just takes it and that's the end of it. There's interaction.
Yeah, the interest can't be skin deep is basically my point.
Yeah. Okay. I want you to listen to some sounds and tell me what words you think they represent. Okay, ready for this? Okay.
Wait, what? Can I get it one more time?
Wait, are we like? Is this like charades but with sounds?
It's like with sounds. It is sound charades.
This is one of those things that we won't be able to know what it is until we're primed, and then we'll be like, oh, we know what it is, and then they go, oh, well, actually, it's this, but you were primed the wrong way. It's like one of those psychological.
Sounds like a can opening.
Okay, so you've got a can opening. All right. Any guess, Justin?
Checkers? I don't know.
Checkers. All right, we're going to move on to the next sound.
That's definitely a can opening.
Yeah.
No. What is that? That's somebody saying "rawr."
Yeah. So I could go really literal and say like tiger, or I could say fierce.
Okay. Ooh. Good. Okay, we'll go to the next sound.
I hate that. So my word is hate.
Oh, are we just supposed to associate a word with it?
Yeah, what does this word? It's word charades.
God.
This sound. What word is this?
No, stop it. Don't do it again.
Wait, 3 more times? Just because I didn't hear it that time, because somebody was screaming over it. Blooper.
Blooper. Okay, we got a blooper from Justin and hate from Blair. And the final sound.
Ha ha. Ha ha.
Hahing.
Hooray.
It's hahing.
So Blair says hooray. What is... Okay.
Hahing.
I will not keep anyone waiting any longer for the answers here. The first sound is meant to represent the word cut. So can opener was very close.
All right.
Cut.
I see what's it. I needed an example. Now I'm off.
Yeah, Blair.
Very well done.
You totally nailed tiger.
Oh, good.
Nailed it.
Tiger.
Nailed tiger. That was that sound. The second sound was tiger. The third sound that Blair hated was water.
That's not water. That's like gloop.
Yeah.
Supposed to be water.
It's like bubbling mud.
And the last sound
Ha
was good. It was supposed to represent the word good. The reason I'm bringing all of these sounds is that researchers from the University of Birmingham and the Leibniz Center General Linguistics in Berlin discovered that iconic vocalizations, so sounds, not words per se, but sounds similar to body movements that have that represent words and have gone on to create sign languages and even languages themselves, that there are iconic vocalizations that convey a wide range of meanings. And they used sounds like this for these vocalizations for 30 different words, different meanings. that are common across different languages. And they looked into people across 28 languages, 12 language families, groups from oral cultures, like speakers from the Palikir living in the Amazon forest, speakers of the Daki in the South Pacific island of Vanuatu. There were a number of people, and they say that listeners from each language were more accurate than chance at guessing the intended referent of the vocalizations for each of the meanings tested. Now, in the test, it was a bit easier than what I just played, just playing the songs, well, not songs, but the sounds at you. They were played the sounds, and then they could choose a word from a list of 5.
Mm-hmm.
So they had a 1 in 20, a 1 in 5 chance of getting it correct. And so, but they picked across languages, universally these vocalizations seem to underpin meanings. And so the researchers suggest that their findings challenge the idea that vocalizations have limited potential for iconic representation, demonstrating that in the absence of words, people can use vocalizations to communicate a variety of meanings. I mean, we've got sounds like, huh? Meh.
Yeah.
Yay!
Yeah.
But
Aww.
but I think it's a very specific room full of nerds. We're gonna get.
I feel like you could say that about
happy.
literally every story on this show.
That happy is not that one I didn't buy for sure.
That guy was happy for sure.
I'm confused.
He wasn't happy. It was good. It was like
good. It was good.
Okay,
good.
it was just good.
Yeah.
Right.
Woo.
Anyway,
There you
it's
go.
a lot of fun.
Exactly,
Potential
that's it.
for
Woo.
vocalizations, sounds
Iconic
to
sounds.
have predated and maybe been ancestral, probably in combination with body movements to represent ideas. There were probably vocalizations and movements, physical and auditory charades that were played by early people to create languages, to create communication that was shared across individuals. So it's just a lot of fun to be able to play this kind of game.
Well, and think about communicating with somebody who doesn't speak your language. They're talking at you in their language, or you're talking at them in your language, and you can often get meaning across with the combination of inflection and body movements. And inflection is at the basis of what this is about. It's about sounds that convey meaning, even if you don't understand, if it's gibberish to somebody.
Yeah, the words themselves could mean nothing, but the way it's conveyed could get the entire idea across. Yeah. Sounds. It's very, very interesting.
All right, somebody is. Hey, is that Blair? Are you wrestling your mic?
Oh yeah, I am.
Okay,
Sorry.
okay.
I was touching it. I'm sorry.
I didn't hear that. This is TWIS. If you just tuned in, thank you for listening. If you've been listening, thank you for listening. I do hope that you're enjoying the show. And if you are enjoying it, please share it with a friend. All right, everyone, time for the quick COVID update for the week. This week, the CDC is kind of going through it after they completely switched directions on their mask guidance. Not 1 week prior, the head of the CDC had been in front of Congress saying, we got to wear masks. Masks are important. It's really good. And a week later, the CDC changed its guidance to say that vaccinated individuals can go without masks indoors and outdoors, and except in places like hospitals, healthcare locations, or on public transit, public transportation.
Or any private place that's still saying to put your mask on.
Yeah, but what it is the hornet's nest is basically that it punted the whole situation onto business owners to decide whether or not to check for vaccination, whether or not people have been vaccinated upon entry. But now we're getting to a point where people are required in some locations to wear masks to enter indoors locations. It's great. Wear your mask. If you're vaccinated, just wear your mask. Or if you have your card and you can show that you're vaccinated, if you can prove it, do that. If you feel comfortable not wearing a mask and you're happy doing that. But I mean, don't make a big deal out of it. Let's just try and make it easy on everybody.
I think the real issue is that 30-something% of the United States is vaccinated. So
we're at 37% of the United States that are vaccinated. And that I do think you're right, Blair. I think that was one of the other issues, is that just the week prior, she had been saying that that we would continue to wear masks until vaccination rates were widespread. Everybody, we're starting to head toward higher vaccination rates, lower case rates. And it seems as though they've just given up. They had said that they were going to wait until we were below 1,000 cases a day across the country. And they've just completely given up on that.
Also knowing that we're about to hit the summer craziness again. And if we look at, I realize people are vaccinated out there, which is great. But if we look at anything like infection trends from last summer, we are about to go into a period that could really be problematic. So I'm not thrilled. The timing seems less than perfect.
Yeah, and from a science brief from the CDC themselves, they reference a study that says, in one study, complete relaxation of prevention measures prior to adequate vaccination coverage resulted in essentially no reductions in SARS-CoV-2 infections. And this was a study in which they simulated various vaccination rates and different non-pharmaceutical interventions, mask wearing, social distancing, all that kind of stuff. And they adjusted the percentage of these different things. And really, in their simulations, not real world, just a simulated study, if vaccination was not adequate and you relax the prevention measures, the SARS-CoV-2 infections stay high. They don't get reduced. It's a combination of things we have to deal with.
Absolutely. Right.
So, yeah. So, to be safe, when you're around other vaccinated individuals, you can be safe without a mask. But if you're around groups, crowds where you don't know the status of everybody else, you don't know if you have immunocompromised individuals, kids under the age of 12 who are not yet vaccinated, all sorts of people who are vaccinated, but for whatever reason, their immune system is not taking the vaccine as well. People can still be at risk. So let's all just calm it down and be nice to each other. Wear masks in public.
This is why what they just did was exceedingly dangerous, because what was said was that if you're completely vaccinated and don't have COVID, you can be with somebody else who's completely vaccinated and doesn't have COVID. But nothing else was said. And the way it's heard is masks off, business as usual.
Everybody.
Send the workers back to the front line dealing with the indiscriminate public, because we're done having. So, hey, let's all have another wave. Let's have another spike. Let's fill those hospitals with more sick people, and let's have a lot more death, because gosh darn it, we have to open the kiosks.
And don't forget, the communication, the science communication around this was poor. Absolutely. Yeah. And we have to remember that there are new variants that are more transmissible. We talked last week about how they're more transmissible in kids and who are not vaccinated yet.
No, they're not even allowed to be vaccinated. Yeah. They're not even allowed to get vaccinated, the children. Yeah. And so you just don't care, is what you're saying. You just don't care about them.
I care. I'm going to wear my mask in a store.
I do too. And please remember this generation that's going to be deciding the fate of old people. whether or not Social Security, do we think we need that? You know what? I kind of recall a time when I was a child and not able to be vaccinated when they were like, who cares about the pandemic? You know what? Bye-bye. Because that's what I think that's totally fair. They should just off anybody of a certain age who's like, we're going to get vaccinated and skip the children. That generation should just absolutely commit parenticide for the entire generation that came before. I totally am for that.
Yes. Well, let's wear our masks. Let's do it for the kids. Another study. So we do know that the CDC is frustrating everyone. Guidance is not great. And even Fauci came out this weekend and said, be prepared for us to update our guidance, because they will probably spend a week figuring out why everybody's frustrated and then make up some new rules. Because, I mean, you hope that our governmental public health institutions have things in control, but everybody is still playing the new pandemic game, the we've never done this before pandemic game, and there are a lot of mistakes, and it's really unfortunate. It's really unfortunate because lives are at stake. Yeah. Another study, in contrast to a study that I reported on last week that posited SARS-CoV-2 viral integration into human DNA, there's a new study out this week in the Journal of Virology suggesting that SARS-CoV-2 cannot integrate itself into human DNA. So now we have one study which has gone through bigger reviews, revisions, that says, yes, SARS-CoV-2 is a retrovirus getting into your DNA. And then another study saying, nope, that is just an artifact from the way that the study was run, and the question still remains. So I put it to the virologists and the molecular chemists and molecular biologists to please figure this one out, because it's an interesting question. Is SARS capable, are coronaviruses capable of hijacking cellular machinery to integrate bits of themselves into our DNA? Is it possible? Big question. And Justin, you have an interesting question as well that you wanted to talk about.
Oh, yeah. So there's 18 scientists from research institutions that are now urging a deeper dive into the origins of the coronavirus. They published last week in the journal Science, and they're arguing that there's not yet enough evidence to rule out the possibility that SARS-CoV-2 virus has escaped from a lab. Why is this important? Because there's a report from the World Health Organization that came out that said it definitely couldn't possibly, no way that it came from a lab in China. Problem is, it was pretty much based on data that was being offered by essentially the Chinese government. So you have to take with a grain of salt any information that's coming from China about this, of course, because all information that comes from China has to be approved by the Chinese state, and if not, they will literally shoot the messenger of that information. And these researchers have come out from various institutions and said, hold up, wait. This is David Railman, Dr. David Railman, professor of microbiology and immunology at Stanford University, who's one of the signers of the paper. We believe this question deserves fair and thorough science-based investigation, and that any subsequent judgment should be made on the data available. Also stating that they and their colleagues have looked at this, and even though that they're like experts in their fields, they don't believe there was enough information or data in this report from the World Health Organization that could actually come to the determinations that the report came to. Within that report, they found that the the chance that it was an accidental laboratory leak was deemed extremely unlikely. The likelihood that the virus jumped from a source animal to an intermediary species and then to humans was considered likely to very likely. Yeah. Also, this was sort of interesting. Other pathways that were investigated, direct jump from animal to human without the intermediate host was considered possible to likely. Transmission from the surface of frozen food products was possible under this study. Yeah, frozen food products. First of all, what are you freezing that you think could have had COVID virus? And is that really higher possibility than
COVID crunchies? Don't you eat those?
Than a lab. Oh gosh. Ravinda Gupta, professor of clinical microbiology, University of Cambridge, who's also a signer of the letter, said he would like to review lab notes from scientists working at Wuhan Institute of Virology. This is a research center where the coronaviruses are studied. It's in Wuhan, which is why there's a pretty decent interest in that lab as being a potential source of its exposure to humans. I'd also like to see the list of the viruses that have been used in the Institute over a 5-year period, something that has apparently not been made transparent, which is a thing that you would think would be curious. It's a point of interest at this point. Tedros Adhanom Ghebreyesus, Director General of the World Health Organization, actually expressed a similar opinion when the report was released, saying, although the team has concluded that a laboratory leak is the least likely hypothesis, this requires further investigation, potentially with additional missions involving specialist experts, which I am ready to deploy. Let me say clearly that, as far as the World Health Organization is concerned, all hypotheses remain on the table. So even though the World Health Organization sort of put out this report based on Chinese scientists provided data, they still haven't completely signed off on it being the end-all, be-all of the situation. Now, another thing to keep in mind, too, is while this is an ongoing thing and we don't know, one of the things that we have not found is any animal source. This is
right.
There were something like, I got it in here, something 80,000 wildlife, livestock, and poultry samples have been collected from 31 provinces in China. None of them turned up a SARS-CoV-2 antibody or snippet of the virus's genetic material before or after the outbreak. So thus far, after what sounds like a pretty extensive search so far, could have been missed. It's still out there. We could easily have missed it. But having looked at that and not found it, it makes you think there might be another source. If it was collected in a lab, if it's in this lab, it could have exposure to humans over a prolonged period of time of working with virus that might otherwise not have been something that was infecting humans right away. It could have been something that was an exposure over time. And any lab keeping a virus in it is very difficult. They're tricky. They can sneak a ride on humans if the PPE isn't perfect.
And part of coronavirus studies is that they're usually not BSL level 4. They're not biosafety level 4. They're which is the highest level of sanitary, keep it in a bubble kind of protections. Usually they're level 2 or 3. So there is a little bit more. There's a bit more relaxation on how techniques are done, the safety precautions that are in play. And they have had viruses escape or go out previously. There have been American labs that have had viruses go out with people from BSL level 2 and 3 labs in the past. So it's not to say that it hasn't happened ever, that things have left the lab. What we're concerned about is gain of function research, whether or not that was ongoing. Doesn't appear that was happening necessarily at the Wuhan lab, where And I've also read different statements that it doesn't that in the scientist notes that have been provided, the communications that have happened, there isn't evidence of this exact strain of a SARS coronavirus being in the laboratory. Like, that's not something that they were working on. So it's a big question. Where is it if it was in the natural population? If we want to find it out in the wild, where's that reservoir? Where are we going to find it? What animal species did it come from? We have no idea. If it was from a lab, there's still, there are outstanding questions. At this point, I think I've seen more evidence to the contrary, but
how?
That it's similar to what the WHO director said, the general director, where the report found it to be like the least likely option. It's still, it's science. So it's like, this is the least likely scenario, but we don't have all the evidence to concretely say it didn't come from the lab. There's still, there are some possibilities, but most of the evidence suggests it did not. So, and anyway, yeah.
I don't know that that's true at all.
But we should researchers should continue to investigate the question of the source, because it will help us understand this whole pandemic and SARS coronaviruses even further. And that on its own is important if it can protect people moving forward.
And I just want to clarify too that the 18 research scientists from universities across the world, including Stanford and Cambridge in these examples here, are saying, no, you cannot say it is the least likely. You do not have data that defends that point of view.
Out of all of the options, they do have data that suggests it's the least likely.
No, they're saying the opposite. They're saying,
yes.
Wait,
I think it needs to be.
Are we cross-talking? They're saying the data that was represented that was used to state that it was the least likely does not support it being least likely or most likely or any likely. They're saying you're not working with data that reflects your outcome at all from the World Health Organization report. They're saying that report is junk and we need an actual investigation.
There were issues with the investigation because they were definitely handled by the Chinese government. They weren't allowed into certain places. They didn't get the WHO, the group of investigators who went to China to put this report together. They did not get to see everything. And that is something that was made apparent very soon after the end of that investigation. So,
and I think that what the World Health Organization director said wasn't that he's agreeing that it's the least likely. He's saying that even though the report is stating this, we still need a proper investigation as well. He seems to also be stating that don't put a whole lot of weight on this one report that was generated off of potentially biased data. That's all.
Yeah, well, and how about we just put any lab with coronaviruses in them at the highest level of biosecurity from this point forward?
Sounds like a great idea.
Well, any lab that's doing gain of function research for sure, mixing different variants together.
And no, that's not saying it's somebody's like, does this mean COVID-19 is a bioweapon? No, it's not that it's a bioweapon. It's just, it's not that it's engineered either, because that's been looked at, and that doesn't seem to be the case at all. What it means, though, is that if you're going to work with things that are potentially dangerous biologically to the world and creating a pandemic, you need to use the greatest security. And even then, even a level 4 can have a breach. Mm-hmm. So it's none of it is, but the lack of transparency, if that is what is taking place, which has, it's been a problem. Hey, we had New York City had a lack of transparency about the COVID outbreaks. Okay. Yeah. Like everywhere is doing, as was like a lot of politicians are hiding things because they want to look better or look like they're handling things better, which just makes the problems worse, which is why we need absolute transparency in these things. And we need a decent investigation because we still don't know where it came. Thank you, Sadie. We still don't know where it came from.
Yeah, it's true. We do. Yeah, exactly. We still don't know where it came from. We need to figure that out. And that I absolutely agree on that for sure. That does it for our COVID update. This is This Week in Science. If you are enjoying the show, please consider supporting the show. You can find our Patreon link at twis.org. Click on that link and choose your level of support over in our Patreon community. $10 a month and up. And we will thank you by name at the end of the show. There are also other fun rewards for being patrons. We can't do this show without your help. So thank you so much for all of your support. It's time now for the wonderful animal breathing, air-filled portion of the show. Blair's Animal Corner.
With Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. There's Animal Corner.
Blair.
How much do you like to breathe?
Oh, all the way, all the time.
Everyone can take
respiration.
A deep breath.
I love it. I don't like it when my sinuses are full.
Take a deep breath in. And out. Doesn't that feel nice?
Yes.
What if you could breathe another way? So this is
suddenly an episode of South Park.
Yes.
What?
So this is a piece of research looking at the delivery of oxygen gas or oxygenated liquid through the rectum. This is a look at artificial respiration, artificial respiratory support. We already use it in other ways, like you get intubated and stuff like that, as clinical management of respiratory failure, which could be as a result of pneumonia, acute respiratory distress, any of those things related to COVID, for example. And so the ability to go in and medically assist with respiration is very important. So Takanori Takebe of Tokyo Medical and Dental University and the Cincinnati Children's Hospital Medical Center wanted to look at using the rectum for artificial respiratory support. The inspiration for this come from sea cucumbers, freshwater fish called loaches, certain freshwater catfish. All these animals use unusual forms of respiration, many of them their intestines. It's a very absorbent surface. And so the idea here was to provide evidence for intestinal breathing in a mammal so that we could try to kind of translate that into human medicine. So this is looking go ahead.
I just need to jump in here and comment on the use of the word breathing.
Yes.
Because when you think
fair
of
point.
breathing, it is the air, your lungs inflating and deflating. Breathing is your respiratory system process. It's not specifically just respiration. Respiration is a metabolic process, which is the transfer of oxygen and carbon dioxide. Your cells respire, right?
Very good point. I used breathing for comical effect, but I think it is very important.
Everybody's using breathing in the headlines because it's like what people understand, and I get that.
The lungs are not involved. There are no alveoli used in these processes.
And this is not allowing animals or people to breathe out their butts.
Right. Unlike turtles we've talked about on this show before.
Yes,
they can breathe through their butts because it is a passive thing that is just part of their normal processes. And so that is what's happening in the sea cucumbers and these fish is they are breathing through their intestines because they're in water. They live in this stuff. And they are saturated by it. And so they can pull oxygen out of the water. So even then, it's not breathing. It's respiration. But that's not even what this is about. This is a step even removed from that. That's rescue respiratory assistance. So there's a difference from that. So we have breathing, which is using the lungs, and you're using that to pull oxygen out of air and push it straight into your... Okay, so that's all happening. Then there's respiration, which can be done by pulling oxygen out of water through something that is not a gill or a lung. In this case, you're just using membranes through the intestine. And then there's this third thing that's even a step removed, that is a kind of manual... how do I put this without it sounding really upsetting? There's no way. Forced contact with oxygen in the rectum to aid someone who does not have good oxygen levels. So here's the actual study. They designed an intestinal gas ventilation system to administer pure oxygen into the rectum of mice. So they're just putting air up there. Without the system, no mice survived 11 minutes in extremely low oxygen conditions. They all fell short of 11 minutes. But when they were ventilated rectally, 75% of mice survived 50 minutes in normally lethal low-level oxygen conditions. So that's huge. So that's step one. So that's just air, right? But this is not going to fly for human medical care because intestinal gas ventilation requires abrasion of intestinal mucosa. If you're not a doctor, you can tell that sounds bad. You don't want that. So it's not clinically feasible. It's not good to push a bunch of air up there. It's not what it wants. It's not good for the bacteria that lives there. It's not
abrasion. You're just disturbing the mucosal lining. Yeah, it's going to mess everything up.
It's bad. So instead, the researchers wanted to look at a liquid-based alternative. So they used oxygenated perfluorochemicals. They've already been used in other medical processes in humans. So they're already biocompatible. They're already clinically approved. So they used intestinal liquid ventilation in rodents and pigs via this liquid perfluorochemical. They received, when they received this intestinal ventilation, they could walk farther in a 10% oxygen chamber, and more oxygen reached their heart. So also their skin looked better. They were less cold. Their skin had more oxygen in it. They didn't see any side effects. So this all looks good. So the next plan is to expand their preclinical studies to pursue regulatory steps to kind of figure out what potential side effects there could be and how they can do this in the best way possible in order to move to clinical translation. So the lead researcher, Takebe, says the level of arterial oxygenation provided by our ventilation system, if scaled for human application, is likely sufficient to treat patients with severe respiratory failure, potentially providing life-saving oxygenation. So if, for example, if you have pneumonia, if you have pneumonia, or if you have a collapsed lung, if your lungs are not working for whatever reason, intubation is not really going to help you. So how can you get oxygen to a person whose lungs are not currently working? This is how. And so this study might seem very silly, talking about breathing through your rear and all this kind of stuff, but really, this could be life and death for people in hospital situations.
Yeah. Like when we determined, didn't we, that the reason people yawn is when they're having an oxygen deficit?
No, because last week we talked about how it's actually about cooling the brain.
Or cooling. Okay, that's right. Cooling the brain.
Yeah.
It's not about oxygen at all. It's not about intake.
Mhm,
Okay.
mhm, 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 mhm.
Okay.
Yeah. So anyway, it could be someday that somebody goes to the hospital and their life is saved by an oxygen.
I think
Delivery system.
the idea behind this is really quite interesting. The idea that by supplementing the body through different methods, where if the lungs, that respiratory surface is taxed, maybe because you've got a virus that's attacking your alveoli and your membranes, right? Maybe the blood vessels there are inflamed. Maybe for some reason you're having issues getting oxygen transfer at that cellular surface. So you've intubated somebody for ventilation to help them. But if you can help their body systems from a different place, from a different angle, to be able to support the oxygen needs of the body, of the cells to be able to survive, then if that helps, it helps. And so the idea is really a fascinating and exciting idea. I think that it is, it's shocking to people because, and there's a bit of a joke factor to it.
Yeah.
But at the same time, if this, they're doing the animal studies, if they can go through the clinical trials and prove that it really is something that works in human patients, this is something that could make a huge difference in survival.
Listen, if you're in the hospital and you can't breathe, anything goes. And honestly, this could really make the difference.
Yeah. I'm wondering if those perfluorinated compounds they're talking about, they've been used safely in people, or perfluorooxygenated compounds. I wonder if these are compounds. There are oxygenated liquids that have been used previously to submerge animals, humans, in a liquid to get there, and you inhale the liquid into your lungs for oxygen transfer. There are situations where that is proven useful. I wonder if this is the same kind of liquid.
I don't know.
Yeah.
Good question.
Yeah. I don't know. Anyway, it's an interesting story. That's fun.
Yes. So from that, I want to talk about scuba diving lizards.
Of course, because we're going to go. Okay, yes.
Yes. I mean, they're connected. It's all about oxygen. So this is a team of evolutionary biologists from the University of Toronto looking at anoles, type of lizard. They are able to breathe underwater. And it's been observed that these guys can dive down underwater for a really long period of time. And there's been a lot of kind of observational stuff related to that. And we know, for example, that they can dive underwater to avoid predators, and they can remain submerged for as long as 18 minutes, which in the grand scheme of the animal kingdom is not crazy long. But if you think about just a normal terrestrial animal, that's a very long time. So how are they doing that? It looks like, okay.
Sadie wants to know how. How? Ruff.
It looks like they're actually, they make their own scuba apparatus. And by that, I mean, they use their hydrophobic skin to breathe out a little bit out of their nose, and they exhale a little bubble that clings to their skin. They then re-inhale the air and back out and back in and back out and back in. We know the oxygen doesn't all go away the first time you breathe in a breath of air. So there's a reserve of oxygen that they're working off of in that bubble. So they're calling it a scuba situation because it's like a rebreather on a scuba tank, right? And so the way that they checked on this is they're not just blowing bubbles for fun. They measured the oxygen content of the air bubbles, and they found that it did, in fact, decrease over time. So they were rebreathing the air back in and out from this bubble over and over. And of course, this helps them avoid predators longer, right? So this would be an evolutionarily advantageous thing to do. And this all started with just some observations back in 2009 and then later in 2019, but they really wanted to test this. And in the end, they still have some interesting questions. So they have some funny hypotheses for future study, which is really what I thought was interesting about this. Is for one, they think the reason they're so good at this is not just their hydrophobic skin, but also that they think they push out the air from their nasal passages and mouth and windpipe, which don't make contact with their lungs. So they're able to basically use this air that's not getting oxygen pulled from it anyway to be able to push it out with what's in the lungs and mix it all up and pull it back in. So it's less depleted because they have that reserve in the rest of their body. But they also think that it helps them rid their body of carbon dioxide. They have a hypothesis that the carbon dioxide in the bubble from their lungs dissipates into the water, because there have been previous studies that CO2, because it's so soluble in water, and because the level of CO2 in the bubbles is higher than the water, it follows the gradient. And so it actually leaves the bubble and goes into the water. And in turn, they think that it might even act as a gill to absorb water from or oxygen from the water, which is something that aquatic beetles do, by the way. They use little bubbles, they exhale, they have this gas exchange, and then they pull it back in. So they really want to look into this and see how intense this bubble is helping them as a scuba tank.
I find it amazing. I mean, it's one thing for an invertebrate beetle to use a bubble like this for respiration, but for a larger organism, these animals, they're small, but at the same time, this is this has to be a significant bubble and process for them to be able to use it like a rebreather, to be able to swim underwater for such an extended period of time. Like that, this is a big deal. How come I don't know how to breathe a bubble out of my nose that I can rebreathe with?
So admittedly, while I was first reading this story, I kind of like breathed air out into my mouth and then breathed it back in and out and in and out and in. And I don't think it helped me at all.
No.
So
And
I
anyone
must be missing something.
Anyone who has tried to inhale the air out of a balloon and blow it back into a balloon and use a balloon as a rebreather, you find that you pass out fairly quickly. That you very quickly deplete the oxygen in that balloon and fill it with carbon dioxide.
Why is that though? Why are we so much, I guess, better at breathing? I guess because we're so big and metabolically complicated. But yeah, it's pretty wild. And of course, they had to throw in here, the researchers mentioned that it's too early to tell if lizard rebreathing will lead to any particular human innovations, but biomimicry of rebreathing may be an interesting proposition for several fields, including scuba diving rebreathing technology, which motivated our naming of this phenomenon. So yeah, it's possible, but also I think, isn't that kind of what they already do?
Yeah. Yeah.
I guess the part that we would want to key into is the pulling oxygen from water and releasing carbon dioxide. I don't know.
Yeah, that there is a diffusion of oxygen from the water into the bubble, into the body of the animal, right? And the diffusion also of the transfer of carbon dioxide out into the water. And so how do you turn a bubble into a membrane that can make this work? That I don't get that. How do you create the diffusion gradient that allows that to happen, but not in it's extending your lung yet with just this buccal cavity with this?
I think that's the thing I'm most interested in regarding this study, even though that's the thing they haven't actually studied yet. But stay tuned.
Stay tuned.
Maybe around episode 1000, you'll hear all about it.
Stay tuned for more scuba diving lizards on future episodes of This Week in Science. Yes. Oh, and I was thinking, just as a complete aside, about there's this scuba diving lizards and the oxygenation through the posterior. I'm wondering if free divers will take advantage of either of these kinds of research for their free dives.
Yeah, I mean, I'd love to do it. Be like,
Could you
dive down in a pool, say, see you in 10 minutes.
Right? Could you supplement the time that you could stay underwater? You know, they hyperventilate to be able to get oxygen in, blow the carbon dioxide out. They have huge lung capacities. They've also trained so that their actual metabolism is different, so that they have this slower gas exchange. And I'm just wondering if you had the liquid oxygen in your gut, in your colon, if that would allow you to stay underwater longer.
It seems like yes.
It seems like yes. But this is just a question that came to me while I was standing here. All right. It's time for Justin to tell us some science.
Oh, what do you want to know about? Let's see.
You're going to extend the animal corner with some more animal stories.
Yeah,
right?
let's talk horses.
Horse. A horse is a horse, of course.
Yeah, a new study of ancient DNA from horse fossils found in North America and Eurasia shows that horse populations on the two continents remained connected through the Bering Land Bridge, moving back and forth and interbreeding multiple times over hundreds of thousands of years. So the new finding was based on genetic continuity between the horses that died out in North America, which was not very long ago. That's about 11,000 years ago. That's before Egypt. That's before current population of Europe was in Europe. This is time when the Americas were actually still, that Native Americans were populating pretty much both North and South America. 11,000 years ago, North American horse died out and then gets reintroduced later by Europeans again. So this study is in Molecular Ecology. It's available online. Corresponding author Ben Shapiro, professor of ecology and evolutionary biology at UC Santa Cruz, says the results of this paper show the DNA flowed readily between Asia and North America during the ice ages, maintaining physical and evolutionary connectivity between horse populations across the Northern Hemisphere. So this Bering Land Bridge that we sort of have thought about, I always sort of picture it as a series of little islands between Alaska and I guess Siberia in Russia. But it's actually, it's much more expansive than that. From, it gives a couple of landmarks here. From the Lena River in Russia to the Mackenzie River in Canada, there were extensive grasslands between those two places because so much of the water that would have been in the ocean was now in ice on land somewhere with these massive glaciers that had formed. And so there was this place where horses and mammoths and bison and other Pleistocene fauna were just moseying about. And in this area in between, these horses that had gotten separated about 1 million years ago were able to get back together, to breed again, to switch sides, maybe even. Anyway, this result is showing that the North American horse that had gone extinct is maybe a little bit more of a reintroduction to North America than it is an invasive species, perhaps. Anyway, new findings are going to fuel controversy over the management of wild horses in the United States, as descendants of domestic horses are brought over by Europeans. Many people regard wild horses in the United States as an invasive species. Others consider them to be part of the native fauna of North America at this point. But whatever the white people in America decide about what constitutes invasive or not will likely be taken with a grain of salt by people who are actually native to the Americas. And in other news, memory is a weird thing. We all have memory. Yes?
Huh?
Huh? We all have memory.
I don't remember. Do I?
We all
What
have
is
memory of some
memory?
form.
Have I learned anything in my life? Do you have memory?
We all have some form of ability to recall things, but we don't all have the same way of doing it. And we don't really talk about it a whole heck of a lot, about how our memories operate. Have you ever met somebody who has a photographic memory? Either of you?
Yes and no. I have a
Okay.
friend who has a photographic memory for dates. She's able to remember exact dates when things happened,
Okay.
which I've always
So-
found fascinating. Like, how do you do that?
I've met one person who actually has like a freakish photographic memory where they can look at something and recall it days, weeks, a month later, no problem. And the way it works with them is it's like they're taking a snapshot, like a picture, like photographic memory is the exact right way to define this. They're taking a picture of it, and it's not that they're recalling what it said. It's that they can be reading it for the first time a month later because the text is saved, right? The visual is saved in their head, and they might not know the information, but they can read it from the memory.
So
That's
this is all long-term versus short-term memory storage? Is that what you're talking about?
It's just memory. It's just however you want to regurgitate something that you have learned. So anyway, compared to that sort of memory, though, that ability to take a picture of something and recall it at any point later, most of us need a lot more help in remembering.
We need techniques.
Yeah, we need some techniques. So some Australian scientists looked into an ancient Greek technique of memorizing, which I actually heard about for the first time in a Sherlock episode, which is the Mind Palace.
Mm-hmm.
The Mind Palace. And that memorization technique is basically one of taking like a childhood home and placing bits of information around in the mental memory blueprint of that house so that you might associate certain information with certain rooms or areas of that house. Something that's already well imprinted in your mind, the rooms of the house or where the bathroom is or something like this. You can lay other information on top of that and apparently get a pretty good recall out of it. So they were studying that, and they compared it to a memory technique developed by Australian Aboriginal people, which have a very interesting, because they have a word of mouth tradition, an oral history. So they have to pass everything down over 50,000 years through that oral history. So memory becomes very, very important. Got to remember the stories of the ancients in order to retell them so the next generation can hear them, that sort of thing. And what they do is actually very similar to that mind memory palace technique, where they have areas of the terrain, areas of the land associated with the information. But then they also take an extra step and they build a little story around it to go with it. So they tested these two techniques, as well as they had a third group of these were all medical students who they were testing these memory techniques on. And they had a third group that basically like watched a video on thumb twiddling. And then they gave them a bunch of information on butterflies. They tried to find something that wasn't, something they were already studying, but they threw a whole bunch of butterfly names at them. And they came back and tested their recall at 10-minute and 30-minute intervals based on techniques. Turns out the Aboriginal technique with the narrative and the sort of locations was the most effective, as folks were 3 times more likely to correctly remember the entire list than they were prior to training on the technique. The students using the memory palace technique were twice as likely to remember the list perfectly if they had than they had before they had studied the memory palace technique. Interestingly, there was about a 50% improvement over the pre-training performance by the group that watched the thumb twiddling video or whatever. So thumb twiddling ain't for nothing, is what I'm trying to say. There's something to that as well. But more importantly, the survey found the students who used the Aboriginal technique also found it the most enjoyable technique for memory.
Yeah, I mean, the memory palace technique, I've always thought that in the memory palace, you're supposed to, you put things in places in your memory palace. So say you choose your house to be the palace, and you go around your house and you put things in a particular order in the house, and you're supposed to also associate them potentially with something kind of bizarre. So that's part of the memory palace technique, where that kind of bizarre association, because of the novelty of it, also allows the item to be associated with the thing that is already in your memory, this very deep memory of a place that you know very, very well. And so that kind of that novelty, that fits really well with this, what you're saying about the Aboriginal memory technique of telling a story. Because along with the story, you're creating context and you're creating novel, unique aspects, details that are going to link all of these items into the locations in the memory palace, in the landscape that are there. So I can see how that added detail, the contextualization, the giving the brain more to chew on as a reason to put things down in memory can help you go through it.
Quick side note to the YouTube moderators: feel free to smash all trolls. The third comment that person had put out, they were already tagged as a troll. Feel free to it's not what the chat room's for. It's not for a conversation for people who are trying to stir up garbage or spout nonsense. Just feel free to boot them. You don't have to give warnings or do any of that stuff. That's obviously somebody trolling. Just boot them immediately when you see that kind of junk. There's no need for that at all. There's
no need, to allow.
Yeah, it's not censorship. You're just not invited anymore. Okay, bye. That's all it is. It's like,
you can go. Leave our party, please.
Yeah.
Just like a person without a mask on can't come into my establishment.
Yeah, no, it's rules, and you're just. It's okay. You don't have to mean to be offensive. It's just if you have a certain smell about you, I don't want to breathe the air around you.
Thanks for saying something,
trolls,
Justin.
Trolls smell bad.
All right, let's dive into some brainy news. Move past the memory palace.
Kiki, yes? you brought a brain story?
I brought a brain story.
What?
That's surprising, isn't it?
Oh my god, it's Kiki's brain corner.
I thought we were taking the week off. I gotta find a Neanderthal story.
I brought one brain story. The thumb brain. Your brain will expand to accept a new thumb. Apparently, researchers looking at a new prosthetic thumb, a robotic thumb that's being developed by researchers at the University College London. They published in Science Robotics about their study to see how this robotic third thumb, how it affects the brain. Because a big question is, how do our brains take in new devices as we learn to use them, and how does our brain change to accept these kinds of devices? So the research started with the design of this quote-unquote third thumb by designer Danny Clode. She designed the third thumb to latch onto your wrist and hand and to, for all intents and purposes, stick out of the other side of your wrist as if it is an additional digit that can be used. And it is controlled by controls on the feet that you can move your feet around to control the movement of the thumb. And it's this really interesting device. They trained a bunch of people on how to use this third thumb to do all sorts of daily tasks, manipulate objects. And in the process, they said, hey, let's put these people in an fMRI and see what happens to the motor cortex of their brain. Usually, before the use of the third thumb, each of the fingers is very distinct in the motor cortex of the brain. It's like there is a spot for your thumb, your first, second, third, fourth, and fifth fingers. So there is a specific spot where the neurons are active to move and use each of your digits in your brain. Upon the integration of the third thumb, this distinction went away and it became more of a finger area in the motor cortex.
Interesting.
So the finger area de-differentiated to a certain extent.
Wow.
Yeah. And it seemed as though even after training had taken place and individuals had stopped using the thumb for a week, that de-differentiation, it stayed in the brain. So the brain
Were they still able to type?
Yeah, they're still able to. Yeah, all the normal stuff is still happening. But it's fascinating that the brain was plastic enough to pick up the new digit. You know, and we pick up new devices all the time. But pencils. We drive cars as extensions of our bodies. We are learning
vacuum cleaner, even.
Vacuum cleaner.
Anything.
Yeah. Anything is an extension of our bodies. We learn how to use them. And we learn how to manipulate them. But in the case of something like a prosthetic, to either replace missing limbs or, in this case, add limbs, add a digit to the body, the question is, if you're adding on, how does the brain deal with that? What if you wanted to have multiple robotic arms that you control with your brain, or that, how does this get integrated? How much can our brains handle? And what's interesting to see is that the brain can handle the extra thumb. It learns how to use it. It integrates it, but there's that plasticity, but at the same time, it affects the way that the brain interacts with all of the fingers. But this was a short period of time.
Right. So yeah, maybe given more time, it could have.
Maybe it would have differentiated back, and maybe it would have added a new digit, a new thumb into the motor cortex. We don't know that.
So you know what it makes me think of is like old school video game controllers, like a Sega Genesis that had like 3 buttons and a directional pad, right? Mm-hmm. And if you were someone who used that and only that, and then you handed them an Xbox controller with like triggers on it and all this different stuff, right? Then I bet you would see similar stuff happening in the brain, where you'd see completely new areas lighting up from when they were using that initial controller. And then eventually it would kind of settle back into the normal video game space, right? Because, I think adding on extra stuff, at that point, it's just a tool, I feel like.
But it's interesting because it is a tool, but so in the case of a video game controller, it's all the motions that your thumbs are normally going to make. It's just learning how to coordinate them with the video game, right? So a different controller, you're just learning that coordination for a different game, different controller. So that's going to be a learning process of just motor coordination. But in the case of like a full additional thumb that you can use in addition to your other thumbs, that's more than just coordinating in a certain extent.
I feel like if this connected to your brain, it would be a better test.
Right. It's not directly connected to the brain in this. Yes,
Because
absolutely.
I think that's the part that I keep getting stuck on, is because it's kind of a pedal based thing.
Yes.
It still feels kind of just like a tool.
Right. I think you're right. Yeah, absolutely. Yep.
So that's good. That makes me think, if I was operating this somehow just with my big toe.
Shouldn't the space in your brain for your big toe become bigger? No.
Yeah, I know. Like, I feel like the big toe, like the fingers could have stayed differentiated. And the big toe's like, I got this. Let me just throw in. And it would be like, anyway. I wonder if my big toe is differentiated from the rest of my toes. I don't feel like the rest of my toes have their own motor.
Your big toe probably is, but the other ones,
the other toes
are
just don't.
Yeah.
It's when the
Toe
doctor
lumps
tells
in
you to
your
wiggle your
brain.
toes, and you're like, "How do I even?"
How do I use these? Yes. Yeah. So it's an interesting study. And I think there's a lot more work that could be done there, because I mean, the future of augmenting the human form we're just getting started with that, and it has the potential to be very interesting, depending on how many of these tools we can integrate. How much can our brain do? How plastic can it be? And what happens? And
like an extra thumb does, though, sound like something that is like a wish of a Neanderthal. It's like an old wish. I don't know that that's as crucial now. Oh, maybe it's becoming
I will tell you, yes,
again,
as someone who has nerve damage in my right hand and drops things all the time, I would love a second thumb on my right hand.
Great.
It would be so helpful.
Would you put it on the same side?
No.
Just to replace the thumb that you have? I was just checking.
No, what am I, a koala?
Do they have two thumbs on one side? Is that a koala thing, really?
Yeah.
Oh, I had no idea.
Yeah.
Oh.
Koalas, they're all thumbs.
They are.
All right, my last story for the night is an interesting story related to the biochemistry of Alzheimer's, and how Alzheimer's and the proteins involved potentially get their start in the neurons in the brain. Researchers at Massachusetts General Hospital were trying to figure out how amyloid beta gets produced. Amyloid beta is one of the proteins that, when it's all gunked up in the neurons, is thought to be a neurotoxin that causes Alzheimer's disease, or at least in part a part of the cause of Alzheimer's disease. They've published in Cell Reports their study looking at the mechanisms of how this all works, and they dug into a bunch of questions related to where in the cell and how does amyloid beta get put together. All right, so first off, a long time ago, this team, the researchers involved, they discovered the first Alzheimer's disease gene called APP. You down at APP? That's amyloid protein precursor. And when this protein gets cleaved or chopped up by enzymes, first beta secretase and then gamma secretase, the byproduct is amyloid beta. So APP is the first step in the chain toward amyloid beta. So they started looking into amyloid, the APP, and trying to figure out, okay, where does APP come from? Well, the lab that was in charge of all this research, they looked at the brain's nerve cells, and in a study using mouse neurons, they were able to determine that there is another form of APP that's called PAL-APP, but it's not friendly. It's not a friendly pal. Mm-hmm. PAL-APP means that this amyloid precursor protein went through a process called palmitoylation. Palmitoylation turned it into PAL-APP. Then PAL-APP gets transported on a raft of lipids. There are lipid rafts in our neurons that are called, they're mitochondria-associated endoplasmic reticulum membranes, MAMs, MAM rafts. These MAM rafts grab up the pal-APP and take it for a ride. And when the rafts take pal-APP for a ride, they go, oh, here's the beta secretase. And then the pal-APP gets all chopped up, and that makes amyloid beta. So in their study, they started blocking or upregulating the MAMs. They got rid of the rafts or they added more rafts, and they found that it totally increased or decreased the production of amyloid beta in these mouse neurons in a dish. So they were able to they were able to use a drug for a protein that's involved, the sigma-1 receptor. So they're drugs that could potentially be used to target a particular receptor that can control the rafts, that can control the production of amyloid beta and stop it from ever happening. So potential intervention for stopping Alzheimer's disease.
Yes, please.
We'll see where it goes.
This show has really made me optimistic about this in particular. I feel like, everyone talks about curing cancer in our time, and there's been interesting strides and stuff, but it really feels like Alzheimer's might get figured out in our lifetime. They're really getting close.
That's not saying much, Blair. Seeing as how...
Oh, because I'm going to live to be 200. I can't live to be 200.
Blair's going to live forever. That's right.
So it's like, wow, in your lifetime. Well, that's not
No, in your lifetime.
Oh, well then, in that case, it better hurry up.
Well, anyway, I love this. I love the idea of everyone I know, and even people I don't know, not getting Alzheimer's. That would be great.
It would be great. Let's keep our memories, not just through memory palaces and storytelling in our brains associated with landscapes. We don't need to. We want to keep our brains working and our memories working at the cellular level as well. Yeah. So you down with APP? No way. No way.
Nope. Get out.
I'm not down with APP.
You're not allowed on the raft, buddy.
Get off my raft. No rafts for you. All right. Have we done it?
We did it.
Oh my goodness, did we?
I think we made it to the end of another show. Yes, we did. And this is Eurovision week. Has nothing to do with science, but I really want to know who you think is going to win Eurovision this year. Everyone, send me your hopes, your votes. I want to know who will win Eurovision 2021.
What is Eurovision?
It's a singing contest.
It's a singing competition. It's
for all of Europe?
All of Europe and Australia.
Okay.
Oh.
Well, there's,
I thought don't the British win that one every year? Is that
No, they lose.
They lose every year. That's the only thing I know about it.
What do you watch it on, Kiki? Not to give free plugs or anything, but what do you watch?
I watch it either on YouTube or through the Eurovision website through a VPN.
Okay.
Yes, I use a VPN.
I'm going to throw in with Buddha's palm in the chat room and say Lithuania.
Norway, says Gorov. Here we go. There we go. It'll be fun. It's a fun weekend. A fun respite from some of our worldly concerns. A song contest.
Did you watch the Netflix movie, and does it reflect what Eurovision is actually like?
It does.
Okay, great, because I haven't seen either. So that'll be my intro.
It's a comedy, but
it
was very, very well done. Okay.
Okay, great.
All right. But on that note, thanks for listening to the show, everyone. I do hope that you enjoyed it. I want to thank my co-hosts for their amazing co-hosting. Thanks so much, Blair and Justin, for another good show. Thank you, Fada, for your help with social media and show notes. Thank you, Gord, for help in our main chat room. Thank you, Identity4, for recording the show. Thank you, Rachel, for all of your assistance. I also want to thank our chat room right now. Our YouTube chat room went through some things this evening. thanks for sticking with us. And together we can all make it through a little bit better. So I'm glad that you stuck with us for this TWIS community. Thanks for being here, everyone, wherever you are. And big thank you to all of our Patreon sponsors. Takes me a minute to share the screen. I'm getting there. Click. I need to figure out how to do this faster. Thank you, John Ratnaswamy, Kira Carl Kornfeld, Melanie Stegman, DeCramster, Karen Tozzi, Woody MS, Andre Bissett, Chris Wozniak, Dave Bunn, Vegard Shefstad, Hal Snyder, Jonathan Stiles, a.k.a. Don Stylo, John Scioli, Guillaume John Lee, Ali Coffin, Maddie Perrin, Gaurav Sharma, Shoe Brew, Darwin Hannon, Donald Mundus, Stephen Alberon, Daryl Myshak, Stu Pollock, Andrew Swanson, Fredis104, Skyluke, Paul Ronovich, Kevin Reardon, Noodles, Jack, Brian Carrington, Matt Bass, Joshua Fury, Sean and Nina Lamb, John McKee, Greg Raleigh, Mark Hessenflow, Gene Tellier, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Dan, Christopher Rappin, Dana Pearson, Richard, Brendan Minnis, Johnny Gridley, Kevin Rails, Back Flying Out, Christopher Dreyer, Mark Mazzaros, Artyom, Greg Briggs, John Atwood. This profile name is hilarious in the context of some other podcast. Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Matt Sutter, Philip Shane, Kurt Larson, Craig Landon, Mountain Sloth, Jim Drapeau, Sarah Chavez, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Stanton, Paul Disney, Patrick Pecorari, Tony Steele, Ulysses Adkins, Brian Condren, and Jason Roberts. Thank you for all of your support on Patreon. And if you are interested in supporting us on Patreon, head over to twis.org and click on that Patreon link on next week's show.
We will be back.
Except Justin won't be.
That won't be. I'll be on an airplane somewhere over the Atlantic Ocean, probably.
Come on, they have Wi-Fi.
Broadcasting live, though, from YouTube and Facebook channels and from twis.org live. The show will continue.
Want to listen to us as a podcast? Maybe go for a nice run while you listen to science? Just search for This Week in Science where podcasts are found. If you enjoyed the show, get your friends to subscribe as well.
For more information on anything you've heard here today, show notes and links to stories will be available on our website, www.twis.org. And you can sign up for a newsletter.
That's right. You can also contact us directly. Email Kirsten at kirsten@thisweekinscience.com, Justin at twistmenina@gmail.com, or me, Blair, at blairbaz@twis.org. Yes, all the domains are different. Just put just be sure to put TWIS, TWIS, in the subject line, or your email will be spam filtered into, let's say, bubble on the nose of an anole, because I didn't want to get gross.
Bubbles are nice.
I took the less obvious choice. All right, moving forward.
You can also ping us up on the Twitter, where we are at TWIS Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world, so I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice, show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand, and all it'll cost you is a couple of grand. 'Cause this week's science is coming your way, so everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause 'Cause it's This Week in Science. This Week in Science. This Week in Science. Science. This Week in Science. This Week in Science. This Week in Science. Science. I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. Science. This Week in Science. This Week in Science. This Week in Science. Science. Got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. So how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you've learned anything from the words that we've said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. Science. This Week in Science. This Week in Science. This Week in Science. Science. This Week in 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 Science.
I love those TWIS leggings.
And we are in the after show.
Has anyone bought them?
I haven't yet.
But have they been bought at all
products.
Do they have any cells? I don't know.
I haven't looked.
I can't talk anymore.
Check. I gotta check out the products. See if they've sold.
That tank top's really cute too.
I know.
If I do say so myself.
Such cute. Look at the Kiwi tank top. It's so adorable. Look at that. I like the back too.
Yeah.
It's got a Kiwi fabric all over it. Kiwis, especially for our friends down under. Yeah. No, for everybody.
Specifically just in New Zealand.
This is the after show, everyone. Thanks. Oh, there's a Justin. Thanks for joining us for the whole episode. And I do want to say, the merch looks awesome, doesn't it, Gaurav? Yeah. Working on. I love the way that the Blair's art in these can lend itself to fabrics, and that can be printed, and that's very fun.
Very pretty.
Yeah. I'm gonna put some of this year's stuff on there pretty soon. So that's a perfect thing for you to email or tweet at me. Is if you want a particular image from this year on a particular item, I will do that.
Yeah, that's great. Yeah. If you want a particular image on your clothings, Blair might be able to make that happen. She is the one. Because as we learned in previous episodes, Dr. Kiki is not the one.
Well, that's
Who
partly
knows how
because
to Zazzle.
I have spent many hours wrestling with said Zazzle. So I figured it out. But
Hey, do they have duvet covers?
duvet covers, I don't know. Let's see.
Yeah.
Products. That's an interesting idea. Let's see.
There's no way.
Because then you can get like
we can make
giant...
duvet. We can make TWIS
They
duvet
do.
covers.
They
do.
have them.
They have
Nice.
them.
They're very expensive.
Oh, I bet. Yeah.
Pretty
They're over
expensive.
$100.
Yeah.
It could be worth it. So I'm thinking either a giant Mastadon one, or just the TWIS logo. Like that would be a little narcissistic of host of a show to sleep in a giant TWIS logo. But I also feel like it's very important to stay themed in life.
I'm gonna sleep in my logo.
Let's see. My products. Create custom products.
There's a lot of things out there that can be printed on. Ooh, what is this? A photo collage fleece blanket. We could make a custom photo collage clock. Square. We can make a TWIS calendar art wall clock.
So here's the question though. Would the mammoth go, because that one's
It would be on the top side, whatever.
well, I mean, he's landscape. So are you putting him, you're not putting him sideways.
Oh, I see what you're saying. No, I'd want, you're right. I'd want it the other way.
I haven't done anything portrait in a long time.
You're right.
Yeah, Melazond, you can absolutely just thrift some bed sheets for your comforter. It is literally 2 sheets sewn together.
But we're gonna print a giant Mastadon on it.
Yes,
Which
I mean
is
there's
different
that.
than
I
just
don't
sheets.
think I can.
So I want it's got to be at least a queen size, which is basically going to be turning. It's eventually going to turn into a square anyway. Big old square.
All right, let me see here.
Come on, get to work.
How about a puzzle? Jigsaw puzzle? Do a puzzle.
Oh, a puzzle. Yeah, that's a really good idea. How did I do it on the? How did I do it on the pillow? This is what I don't know.
No.
This is what I don't know. I gotta tell you.
You have more fun things.
This is what I don't know. Here, do you want to? Here, I can show people what I'm doing if you want.
You can show people what you're doing, but I just found I just found photo sculptures.
Huh?
Photo sculptures, huh?
What?
This could, I don't know why, but this would crack me up. Oh my gosh. Hold on, Blair. I gotta show you what a photo sculpture looks like. Could you imagine?
Let's see.
Our TWIS team photo. That's a photo sculpture.
Sadie, get off the desk. Oh my god.
Sadie's on the desk now.
Did you know my dog is a cat?
Yeah. So this is interesting. I actually wanted to do. I found it. There's a place in Canada where you can go. There's probably more than just a place in Canada. This is something I found on the interwebs.
You find it in the interwebs.
Discovered one day on the interwebs that at least at some point there was a place in Canada where you could go and get 3D scanned. Sit in a booth and it 3D scan you, and then they would 3D print a bust of you. You could make your own little sculpture. We could do that. We could do 3D sculptures that we could send out to people then, like bobbleheads. Except I think
need to be bobbleheads if they're anything. It would have to be bobbleheads.
It would have to be bobbleheads.
Yep.
Okay.
It would have to be. Yep.
Justin, do you want this duvet cover or what?
I think that looks awesome.
Oh my god.
That looks super rad.
Oh, it's gonna say like, oh yeah, it's pixelated because it's so big.
Blowing it up too big.
Yeah.
I bet there's a
Yeah, well, let me sell it.
You'd have to up-res the original image to be able to do it. Probably.
I don't even know how to do that.
There are
All
programs.
right, never mind. But that looks awesome. That does look really cool.
$169.
How much?
$169.
Okay, but what
bit.
does a duvet normally cost?
Like $40.
What do
Depends where you're getting it.
what thread count
But
are
it's
you
not going to be
buying?
personalized.
$40.
Yeah, that's true.
Yes. Noodles wants a TWIS puzzle. Wouldn't that be fun? Blair
But they
art.
do puzzles.
Blair art jigsaw puzzle.
I cannot sleep in $40 thread count.
It's like... Well, my
Good
face
night,
never
Fada.
touches the duvet. I don't know. Oh, page unresponsive. All right.
What about the rest of your
Thunder
whole body?
Beaver
that
says,
is
"Go back
touching
to
it?"
your
That's
original
the-
and make an SVG
My body
with
doesn't
Inkscape."
touch. My body doesn't touch the duvet. I don't know what you're talking about.
How do you sleep without touch? You levitate above the.
No,
Don't
it's called
understand.
sheets.
A top sheet. It's called. You have a bottom sheet, a
Oh,
Yeah.
you're a top sheeter.
and a duvet.
Yes.
If I'm top sheeting, it's because that's the only thing I need. It's no other time do I bother with the top sheet.
Why?
So, a
why?
Because you use the duvet cover because you can take it off of the
Yeah, it's a
comforter,
sheet.
the duvet, and
It's a
wash
blanket
it.
in a sheet.
But that's
It's
a
a
huge
blanket in a
pain in the
sheet.
butt.
Yeah. What?
Also,
It's
I
the
really
same.
like that. Like you can have flannel sheets when it's
So you
cold,
don't-
and
you
they're extra
don't-
cozy. And then you can have like
Yeah, I have
linen
this.
sheets when it's warm out, and it's.
But I
it's
put those
breathable.
down. Those
The
go down at the bottom.
science of blankets.
I
Now, let's
just-
go down at the bottom. And now I'm picturing you do not change your duvet covers very often, do you?
I don't have a duvet cover. I have a comforter because I don't. My body doesn't touch my
So
comforter.
it never gets clean
Because she has sheets.
Correct, because I don't. It
Oh,
doesn't.
that's
I
not-
don't touch it.
because you don't touch it because you've got the sheet.
Yeah.
How
You got
often
the
do you
sanitary-
wash the comforter that's inside your duvet, though, Justin?
Here I'm gonna.
Oh, never, because I have
Right.
a duvet cover. That's the whole point of the duvet. Yeah, you're right.
Yes.
Yeah.
There you
He
go.
who lives in a duvet house.
Shouldn't throw stones or what?
Yeah, right.
Oh,
but I
Blair.
mean, if you're in a duvet house, that duvet is going to catch them pretty paddedly.
Oh.
It will. It will.
Let's see. Oh, all of my art from this year is on my Google Drive.
LL Bean has great comforters. They're very nice for cold weather. But I understand what you're saying, Gaurav. Those catalogs.
Oh yeah. I've recently started getting, I think because the internet knows I works for Parks and Rec now, and it also knows I'm in my mid-30s now. It's started sending me LL Bean like ads, just
Huh?
ad nauseum. Just LL Bean and Eddie Bauer is like all I'm getting is my suggested ads. I'm like, my algorithm is very specific.
I live in Portland now.
Oh,
And I'm middle-aged, so they know I'm cold. They're sending me the LL Bean catalog. Yes, they want me to buy turtlenecks and fleeces and boots, and then they're also sending. I've also started getting. I don't know why. It's the Sundance catalog. So it's like a store
Oh.
that is based on like Sundance,
Huh.
Colorado. I don't know, and
Okay.
yeah, it's like yeah. It's
Oh,
very nice looking things.
Yeah. I
But
mean,
country, very
this is
white
part of
country.
the algorithm too, is I belong to both REI and Sports Basement. So
There he is.
Mhm.
it's definitely also part of it. But
Sports Basement because you can get things cheaper.
I love Sports Basement.
Love Sports Basement.
I love it so much. I love the fact that you can buy the last several pairs of Converse I bought were from there, and they were like $30. This is my place to be.
Ooh, a TWIS sleeping bag.
Yeah, I have an ad blocker. So I did the ad blocker plus hot rod like you're talking about, but they recently, I don't know if they were acquired by somebody or something, but now
You get more
If-
ads.
if you pay them, they will block all ads. And if you do not pay them, then they will let ads through from companies that pay them. So I installed Ad Blocker Plus from all my stuff.
That sounds like that's like mafia techniques.
Yeah. Yeah. So
you're not going to give us the money? Then we're going to give it to the one who does give us the money. That's the way it's going to go.
Yeah. Well, plus I ended up having to disable it in a lot of websites. Like basically if I wanted to do any online shopping, because there were weird cookie things happening that weren't. Yeah. So I had it off for most websites anyway.
Nothing greater than cookies for later.
Then golf with a gator.
A ride in an elevator.
That's a child of the 80s and 90s thing. There's nothing greater than golf with a gator. It's gator golf.
What are you talking about? I have no idea what you're talking about. You're just making nonsense sounds.
Okay, hold on a second. I'm screen sharing again.
No clue. These words that Blair is
somewhere there are people who understand what I'm talking about. It was gator golf. Look at him go.
I feel like, like sometimes kids pick up on something and they focus on it, but it's not really shared. It's like nobody else is paying attention to that thing. What is this?
It's gator golf. I have to find. Hold on.
It looks a little bit like it's built off of the Hungry
Here we
Hungry
go.
Hippos
1994. This has got to be it. You're not going to be able to hear it, but. Oh, I need a video. So we won't get kicked off YouTube, right? Because you can't hear it.
I don't know.
It's on YouTube. The thing you're playing is onto YouTube. How can you get in trouble from YouTube if it's onto YouTube?
Yeah, what could be greater than playing a
the
game of golf with a gator?
Golf with a gator. I get it now. Okay.
All right.
All right.
Yeah, that's very specific memory. I don't think that was a big craze or fad. I think you remember this, but I don't think anybody else did.
No, it's like how also I could probably recite the entire ad for the Shane Company from the radio. It's
the
just
Shane Company
in Cupertino and Walnut Creek.
But that's a constant. That's a constant Bay Area commercial.
And online at shaneco.com. Anyway.
Yeah, it's been a while since I listened to KGO. I can tell, but it's bringing back memories.
Yeah. No, this is the gator golf thing is an ongoing joke in our household. Though I have to text him right now that we're talking about that.
It has come up.
Anyway, I'll send this out in Twitter. I feel like I want to hear if people remember this.
I'm sure people do. I have no recollection of golfing with the gator.
Don't think I've ever come across that thing.
Does anyone remember it?
Ever.
No, nobody in the chat room is saying that. Oh, Thunder Beaver's partner knows all about it. There you go. What is this, Garav? I can speak what? Simlish?
The pooter? What? Simlish? Yes, Blair.
What's okay.
Pooter tutorial? What did I just click on?
What did you click on? I didn't send you anything.
No, someone in our chat, WizMike, sharing the pooter.
What is that?
I don't have this sound on. Hold on. Does it make fart noises?
I'm listening. It's
it's a little device that makes farting sounds. And this.
What a weird video. Okay. Anyway.
This mic, that was funny.
I'm trying to. Oh, good. Okay.
Oh, that was
downloaded.
great.
Lovely. Okay, I have... Great. I have my art. All right. I'm going to make a jigsaw puzzle. Here, I can share my screen again. Okay. What should we do? Which one? Maybe the lemur.
Lemur? I haven't done that before.
The lemur.
It doesn't go back.
It was when I was at the zoo, people always would accidentally call them lemurs. Oh man, it's not a lemur.
It's not a lemur.
I'm at the Lemur Cafe. Like, no, you're not. Is the thing. You're not. You're not there. Sorry, bud. Oh, these would be too hard, I feel like. I have to find something that has more colors.
Oh, those would be so hard. Oh my goodness. It's just like
it would be better for one of these, probably.
Here is a blank puzzle with all white pieces.
Yeah.
Or all pieces of the same
one?
color.
They don't have a square puzzle, do they? This is the problem. I keep doing my art in weird shapes. Oh, there's a square.
What's the right shape for art again?
That's a good question.
Nowadays, the right shape for art is the shape that fits on products.
Oh, I hope not.
I'm kidding.
Ooh, one of these might be good. Like, what do we like? What do we like?
Bright colored. Oh, maybe one of the bright colored ones?
The rhinos?
The stained glass ones?
Yeah, the problem with the stained glass is that it's darn square, but I could still fit it.
It's a darn square.
It's darn square. No, it's a darn square. How about, no. What do we want?
Something
You want a turtle?
fun.
No, we don't want a turtle.
So,
How about
Justin,
some
you're coming back next week to the
Yeah.
US of A.
I'll be back in the United States soon. Yeah.
And you'll be traveling on the Wednesday night, Thursday morning.
Yes.
Yes. Okay. Cool. And then you'll be back for a little while? Okay. Any plans we should be aware of?
Any plans? Such as?
I don't know. Going back to school, getting another job. I don't know. Are you making other plans?
Yeah, there's like job
Working
stuff.
on your truck or your
There's job stuff, there's school stuff, there's bus stuff, there's all sorts of other
Yeah,
stuff.
Bus
So
stuff.
Mm-hmm.
You wouldn't
There's some-
understand. It's bus.
No, you wouldn't understand. There's some high priority things, and then there's some other priority things, and then there's also, bus stuff. You have to speak bus language to understand it
You
properly.
have to speak bus. What is this? My dad scanned these, and this one just says fish. There we go.
How come all that fish's eyes are on the same side?
How come your eyes are not? You ever think of that?
Well, both my eyes are on the front side.
Front side, yeah, exactly.
Okay. Whatever. Whatever, buddy.
Bifurcation.
This one's fun.
There for a reason.
That'd be a fun puzzle.
Yeah.
Would anyone want to make this puzzle in the chat room?
Who
Tell me.
wants a fish puzzle?
Fish
What
puzzle?
is that? But that puzzle's not enough pieces. It's like 20 pieces or something. You need like 1,000-piece puzzle to make it interesting.
All right.
It's like a child's puzzle.
Here's a 520-piece puzzle.
It's getting there, but you really got to go
scary.
for more. You need more than that. Now, it's got to be at least 1,000. Otherwise, it's
Really?
there's no point. Yeah.
That's big.
What else should we try?
It's not
Insect. What do I think insect is from the last? Oh, I think I know what it is.
All right.
Yep.
I have to go to wake.
To go be awake for the day.
I have to go be awake for the day now.
Okay, Thunder Beaver likes 500 to 1,000 pieces.
1,000 minimum.
Thunder Beaver says the more difficult, the better.
Yeah.
Okay, how about this one?
And these
Everyone
have
like
the
this
weird
one?
shaped pieces too. You can't just have standard pieces.
Here's this one.
I like that weevil.
Giraffe weevil. Yeah, I think I'll do this one. There's enough stuff on it. Like this area will be really hard.
Yeah.
But this is all going to be easier because of all the lines. So maybe I will sell this one.
There's going to be a lot, but the background behind
Holy.
the weevil, that's going to be hard.
How expensive are puzzles normally?
How much is it?
It's $40. Is that normal?
I don't know what the price
No.
and nothing seems like a normal price to me on anything anymore. So
Well, Zazzle
it might
stuff
be
is always
reasonable.
more expensive because it's the print on the demand stuff.
It might be reasonable. It might not be. It's no way to tell anymore. It's really no way to tell.
252 pieces is $21 less.
Yeah, but anybody who's over the age of 7 is
Oh,
going to be
there-
bored by that.
There is 1,000.
And I'm
piece one,
apologizing
but
right now to 7-year-olds who are probably underwhelmed by a 250 piece puzzle.
There's a 1,000 piece one, 1,014 piece. It's
That's the
$60.
minimum. That's the minimum size for a puzzle.
Identity 4 was a 2-piece.
I would like just one piece puzzle. No, never mind. Just give me the picture. Doing, Cappy?
Yeah,
All right.
I agree, Noodles. That's
Doing
too much
something
money.
down there. Let's see.
Oh, maybe
Anything
I'll sell
else that we
this
need
one.
to talk to?
$18.
Talk
seems
about?
reasonable.
No, Justin will be back in this
Not next
in this
week, but the
time
following
zone.
week.
the following week. So maybe we can start planning ahead for movie time.
Oh.
Oh, yeah.
We will be in the same time zone. It'd
Mm-hmm.
be fun to do that. And
Mhm.
like, yeah, I'd love to brainstorm
Yeah.
movies that we could watch. And I'd love to, we did a bit of brainstorming last show.
Mhm.
I also would love to get brainstorm ideas from both of you on guests that we should invite. If you have thoughts.
Oh, gosh. Ooh, yes. I
I
had one a little while
do.
ago. So
I do tend to like guests who are in kind of North America time zones because other time zones are difficult. I don't know, how many people want to get up super early in the morning like you, Justin, and speak with us. If they're down for it, that's great. But I think figuring out time zones is also interesting. But yes. Thoughts. Some polls for movies. I can do that. I can poll for movies.
I also think that, Kiki, when you find it convenient to come down to the Bay Area,
Yeah.
we should all be in the same space since we're all vaccinated at the same time. That would be fun.
That would be a lot of fun.
Could do a live viewing of a movie.
Oh, yeah. That's still possible, huh?
That could be. That could happen. Yes. Yeah, TWIS starts pretty late. We've had a lot of East Coast guests, but they don't want to hang out after their interview.
Ooh.
Usually at Identity 4, they're like, the East Coasters are usually like, "Hey, that was a lot of fun. I'm gonna go to bed now. It's midnight."
So that's why if we could get some of the Europeans Central Time to get up early enough to do the show,
Yeah.
then it's still like too early to do anything for quite a while.
Yeah.
So it's not like they have anywhere to go. Nobody's even going to work at this gosh awful early hour in the morning. People are just now starting to wake up
Yeah.
in Central European time.
I think we should watch Mars Attacks, guys.
That's what you said last week.
I
love
know.
Mars Attacks.
I think we should.
I really like all the Men in Black movies.
Oh,
Yeah.
so Brian was just saying yesterday he wanted to rewatch those movies.
Love them. Love those.
So that would be great.
So good. Yes, Stride. Talking is hard early in the morning. It is difficult. Justin can do it. Blair can do it.
Mm-hmm.
I cannot. I love. Yeah, live viewing outside. That's always fun. Whiz Mike, all capped. Yes, Men in Black. Okay. I have
Great.
support for
I
Men in
like
Black.
this. Yeah.
Dr. Becky Smethurst. She's in the UK. I don't know. I've been fiddling around with the idea of having like a TWIS interview separate show where it'd be a different time of day for people in different time zones. I don't know if that would be possible for both of you though.
Say it again?
So
What? Yeah.
So-
what time of day?
yeah,
Right.
I mean it would be like 8 or 9 in the morning to handle time zones for people who are in the UK or
So depending on the day, I
Yeah.
could definitely do that. Depending on the day, I could do almost any time.
okay. Right, because you
It
work
just
from home.
really, yeah. Well, and not for long because they just lifted the telework whenever possible restrictions.
Mhm.
So we'll see. But I'm salaried, so that does mean that my work day is flexible sometimes. So I can.
Hot Rod, are you suggesting that we would all hang out and chat in Discord while we watch a movie together? I was thinking that we would do a watch party. I don't know. We would be on Twitch or something. I don't know. We could talk about the technicals. If you all have ideas on the technicals of a watch party.
Well, I think
Let me know.
the nice thing about doing it on
Gypsy's
Discord is that you're not talking over people's movies. You're just typing.
Right. Well, it would be the same thing for like Twitch too.
Oh, you do it on Twitch that way too. Yeah.
Yeah. It would be
Yeah.
we would all start the movie in our separate locations
Yeah.
at the same time and then
We could even do a chatty beginning, and then we could also have an intermission if we wanted, and then have a post or just a pre-post or
Mhm.
or none of it.
Do I get to eat red vines? I'm in for all of it as long as I get to eat red vines.
Or none of it.
I must have the red vines. Yes.
Okay. All right. I
Chicken
gotta
Hearts
go.
puzzle. Okay, go do daytime things. We should
I'll do-
probably head out as well.
Yeah.
It's 10:40.
Bedtime.
Bedtime for bonzos.
Mm-hmm.
Everybody watch Eurovision if you can. You're in Europe. You can watch it, Justin.
Ooh.
You can watch
Ah,
Eurovision
why?
on
I don't
Saturday.
have-
The finals
See-
are on Saturday.
Unfortunately, I don't have a television. It's so sad.
You have a
But-
computer.
Yeah, but I don't use it for that
Oh,
stuff. So
okay.
it's okay.
You're not interested in the popular media. I see.
No, it's because it's poison for brains. That's why.
What TV in general? Is that what you're saying?
Yeah,
You sound
bad
like my.
for brains.
you
If you
sound-
watch too much television, your brain turns to mush and it oozes out of your ear.
Okay, now you're sounding like my dad. I think I heard that a lot of times growing up.
Yeah, I did. I heard that a lot too. Yeah,
Hmm.
brain's gonna turn to mush. Come out.
Not if you're
falling,
watching
rolling,
This Week
oozing
in
out of your ear.
Science.
No, if you're listening to this quality programming, your brain is actually improving in its killing power. Not
Okay,
you
so Dr. Becky Smethurst. What's her name?
sounded right.
Smethurst. Okay, I gotta find her. I just have to write it down in the note pile. That's what I need to do.
Okay.
Got note
Good morning,
piles.
everybody.
Are we ready? We're going to say things. Yeah.
Oh,
okay.
are we going to say more things? Okay. Hey, say goodnight, Blair.
Good night, Blair. Say goodnight, Justin.
I can't. Oh, I can't.
You can say goodnight to me. That would be the polite thing to do.
Okay.
But then he would say goodnight, Blair. He has to say good morning, Justin. Say good morning, Justin.
Good morning, Justin. Because it's from the burns. Anyway.
Good night,
Kiki.
Kiki.
Good night, everyone. Thank you for joining us for another episode of the show. Thank you for being kind and compassionate and exploring ideas, being curious, and allowing other viewpoints, but also getting rid of the ones that are trollish.
Never tolerate trolls.
Never tolerate the trolls, but let's talk about different ideas. That's what we can do here. Let's discuss, but not tolerate aggressive behavior.
Tolerating fools.
Really appreciate you all so much. Thank you for joining us for another episode of TWIS. Have a wonderful night, morning, day, wherever you are, and we'll see you again next week.